Welcome. Let's look at the third week of embryonic development. If you have not watched the video on the first and the second week, please kindly go and do so as to align with this lecture. The third week of embryonic development are events that occur between the 15th and the 21st day after fertilization. And if we go back to the second week of embryonic development, we know that we have the establishment of the bilaminar germ disk, which is the formation of two germ layers, which are the epiblast and the hypoblast. So this is the configuration that we finally arrive at during the second week of embryonic development. Now, going through the third week of development, there are a number of events that will occur within the developing embryo, and this is what this lecture will unfold. And the effects of different activities that will occur during the third week of development is to create first body alignment. So we have the craniocaudal establishment of the body, and also the symmetrical alignment into the right and left part of the body will also be established during the third week of development. It is during this phase of development that we'll be able to deduce where the cranial and the caudal region of the embryo will be, and also it's going to help to create a form of bilateral symmetrical arrangement of the body, so that what you have on the right side will also be what we see on the left side. So this is where these characteristics are established. And also the other effects that we would see as a result of developmental process of the third week of embryonic development is the formation of the primitive streak, and this will lead to gastrulation. Gastrulation is the creation of three germ layers. Then after gastrulation, we have the initiation of neurulation. We have activity that will occur that will help to induce the process of neurulation. And we know that neurulation is the development of the nervous system. So let's go through this lecture to see how these activities are unfolded and also the basics that are involved in them. Let's check out the alignment. This is the bilaminar germ disk, which is made up of two layers. This is the epiblast, highlighted in red, and underlining it, we have the hypoblast, highlighted in blue. This region, above the bilaminar germ disk, we have the amniotic cavity, and below it, we have the secondary yolk sac. There are different activities that will occur within the bilaminar germ disk, and this activity will tend to occur simultaneously. So as one is occurring at a specific region, the other one is also occurring in another region. So let's go through these to see how these come to play. We have the creation of the precaudal plates. The precaudal plates will help to establish the craniocaudal alignment. The precaudal plate will occur as a result of thickening of the endoderm. Remember, we have our epiblast highlighted in red, and we have the hypoblast highlighted in blue. These are the structures that we got at the end of the second week of embryonic development. So there will be the commencement of a thickening process of the endoderm. So there's going to be a thickening as a result of increased proliferation of cells around this region. So they will begin to proliferate and increase in number, and as a result, they become thickened. As they thicken, they will be pushed to come in contact with the upper cell, and this is what is presented here. And as soon as this configuration is achieved, there's going to be the formation of the precaudal plate. This precaudal plate will help to establish the craniocaudal alignment of the developing embryo. So when this is formed at this phase, we can now say that this region will be the cranial region, while the other end will be the caudal region. So this is how the precaudal plate has helped to establish the craniocaudal arrangement. And so this is the precaudal plate. And also we have the development of the buccopharyngeal membrane and also the cloacal membrane, created at points where the endoderm is in contact with the ectoderm. So we have a point at the cranial region, just anterior to the formation of the precaudal plate, where they form contact with each other. And at this point, we have the creation of the buccopharyngeal membrane or the oropharyngeal membrane. This membrane is the primitive mouth, and it's going to open up to become the mouth as development proceeds. And if you go more cordially, we also have a point where the ectoderm and also the endoderm form a contact point at the cordial region, and this region will be termed the cloacal membrane. This cloacal membrane also is the primitive anus, which means that it will further also develop to become the anus. At this region, we have the buccopharyngeal membrane or the oropharyngeal membrane located anterior to the precaudal plate. Then at the caudal region, we have the creation of the cloacal membrane. We should also know that at the point where we have the formation of the buccopharyngeal membrane and the cloacal membrane, at this region, there is no mesoderm created in this region. It's just a contact point between the superior ectoderm and an inferior endoderm, because we've said that during the 3rd week of development we have the formation of 3 germ layers. So let's see how the mesoderm is able to come into play for it to now be embedded between the ectoderm and the endoderm. But before the formation of the mesoderm, some activities will need to occur before we have the creation of the mesoderm, and that will lead us to the formation of the primitive streak. Remember, we already have the craniocaudal establishment. So upon this establishment, we have the formation of the primitive streak. This primitive streak is formed as a result of thickening of the surface epiblastic cell. And the thickening comes in a specific pattern, seen as a midline thickening of the epiblastic cell. Remember, this surface epithelium is the epiblastic cell, and this is highlighted in black. You can see some of the cells at this region, and also you have the inferior hypoblastic cell at this region. So how they are formed is that the surface epiplastic cell will begin to thicken, and the thickening will begin at the midline region of the epiblast. And that is why you see this red coloration presented here. And this is what the primitive streak is. You can see the midline thickening of the epiblastic cell. This is the surface epiplastic cell, and they begin to thicken around the midline region. And also the thickening will begin to increase and extend towards the cranial region. That means it will extend towards the precaudal plate. This is the precaudal plate in this region, and anterior to the precaudal plate, we have the buccopharyngeal membrane or the oropharyngeal membrane. So as they thicken, they will run forward to the region where the precaudal plate is located. So we have the formation of the primitive streak. So if we look at this image below, we have the primitive streak highlighted here. This is another presentation. This can be related to this. So we say we have the precaudal plate, and this is the precaudal plate in this image. Then anterior to the precaudal plate, we have the buccopharyngeal membrane or the oropharyngeal membrane, and this is the buccopharyngeal membrane that is located anterior to the precaudal plate. You can see it located anterior to it, and also in this image, it is of course located anterior to it. Then more cordially, we have the cloacal membrane, and this is the cloacal membrane. In explaining how the primitive streak emerges, it begins from the caudal region, that is towards the cloacal membrane, and it begins to increase and lengthen cranially towards the region where we have the precaudal plate, and you can see the extension. And we said that this does not end there. If you look at the pattern by which it extends, at the terminal point, it creates a form of rounded eminence that is called the primitive node or the primitive knot. And this is the primitive node or the primitive knot. This is what is presented here. Also in this image down here, you see that it extends from the midline of the epiblastic cell, and it finally ends in a rounded primitive knot. So we have the formation of the primitive streak. The primitive streak now ending as the primitive knot. Of course, the direction is towards the precaudal plate at the cranial region. And what this primitive streak establishes is the symmetrical alignment of the body. So as soon as this primitive streak is established, that ends in the primitive node, there's going to be the establishment of a midline symmetry of the developing embryo. So at this stage, you can say that we have a right part and also the left part. Right and left part that is cut along the midline plane. It's already putting the embryo in the normal phase of life. So we have further transformation of the primitive streak, and this is where it gets more interesting. Remember, we already have the establishment of the primitive streak that ends in a terminal rounded presentation that is called the primitive knot. The next phase of development will be that epiblastic cell that deposited along the midline of the primitive streak and the primitive node will degenerate. There will be loss of cells located within the central part of the primitive streak. This is the primitive streak and this is the primitive node. So there's going to be the degeneration or loss of cell along this central part so that there will be the creation of a groove or a channel through which cells can pass through to go to other regions of the developing embryo. So we now have the formation of the primitive groove and the primitive pit. So this is the primitive groove and this is the primitive pit. So going back to this image, we know that this is a primitive streak because of the creation of groove as a result of loss of cells within the central part. There's going to be the creation of a groove around it. So you now have the primitive streak now being transformed into the primitive groove. And also because of the loss of cell within the central portion of the primitive node, we now have the transformation of the primitive node into the primitive pit. So we now have the creation of a channel through which cells can run through to be able to go to other regions of the developing embryo. And this is the sole essence why this degeneration or loss of cell occur to result into the formation of a groove and a pit. So as soon as this is created, then movement can be initiated. So since a pathway has been created for movement to occur, but the movement will not just occur. The movement occurs as a result of fibroblast growth factor 8. This protein is specifically secreted or released by the epiblastic cell that surrounds the primitive streak. What this does is to loosen the junctional complexes between these cells. If you look at this image, these are the epiblastic cells, and these cells are held together by junctional complexes. This fibroblast growth factor 8 will help to break down these epiblastic cells so that they now have the capacity to move. And that is that they now become very mobile. So they now have the strength to move from this layer and run to different regions of the embryo. And the movement, of course, will occur through two channels. So movement will occur through the primitive groove, which is created along the primitive streak. And another grade of movement will occur through the primitive pit, which is created through the primitive node. And these different grades of movement, they have different specifications, and also they have different structures that they would form as they move. If you look at this image, this is the primitive groove and this is the primitive pit. So you have epiplastic cells. Since they now have the capacity to move, you now see them moving through the primitive groove. You now see another group of cells of the epiblastic layer running through the primitive pit. So there is a specific pattern by which they move. And of course, we'll highlight that as we move through with this lecture and also establish the structures that are formed as a result of this specific movement. This is movement through the primitive groove. Then we have another grade of movement through the primitive pit. Let's go to gastrulation. Gastrulation is the formation of three germ layers. Gastrulation will not occur unless we have the formation of the primitive streak. More specifically, there is a need for the creation of the primitive groove so that the pathway for movement of cells will be created. If this is not created, there is not going to be the path through which the cell will move. And of course, the formation or the establishment of the three germ layers will not occur. So let's look at the processes. We already said that we have the establishment of a groove through which the cells will pass through. The migration that occur along the primitive groove is what we are going to first look into and see what they establish. So cells of the epiblast, since they now have the capacity to move, see them running down and being sandwiched between the epiblast and the hypoblast. And that is where you now have the formation of the three germ layers. The cells that migrate and are sandwiched between the epiblast and the hypoblast will now be termed the mesoderm. At this region, they are specifically referred to as the intraembryonic mesoderm. Remember, we have an extraembryonic mesoderm, which is the mesoderm that is created outside the developing embryo. But because this is created within the embryo, we will now refer to it as the intraembryonic mesoderm. So we have the mesoderm sandwiched between the epiblast and the hypoblast. These cells also further move and they now displace the hypoblast. So as soon as they displace the hypoblast, they also tend to replace the hypoblastic cell. And after this replacement, they are now being termed the endoderm. So that is how we have the establishment of 3 germ layers. It is safe for us to say that the ectoderm, the mesoderm, and the endoderm are of epiblastic origin, because it is the epiblastic cell that runs down to be sandwiched between these 2 layers and also go as far down, displacing the hypoblast to become the endoderm. That is how we have the creation of 3 germ layers. So we have the ectoderm, we have the mesoderm, and we have the endoderm. So at this stage of development, the developing structure is termed the gastrula, because they are now seen with 3 germ layers. It's good to add that in some tests, they would tell that the migrating epiblastic cell will not go beyond the level of the mesoderm. So you just see them being sandwiched between the epiblast and the hypoblast, while the apoblast will then be referred to as the endoderm. So it depends on the text that we are reading and what we want to align with. So in this case, it will then be that the ectoderm and the mesoderm are just the 2 layers that are of epiblastic origin, while the endoderm will be from the hypoblast. Neural induction. This is the next phase that occurred during the 3rd week of development. We have the process of neurulation, which entails the development of the nervous tissue. But we have the induction of this process occurring in the 3rd week of development. So what induces the process of neurulation? So what happens is that we already said that there is movement through the groove that is created within the primitive streak. There is a groove created here, which is called the primitive groove. But what we are going to concentrate more is the groove that is created on the primitive node, and this is called the primitive pit. So we also have group of cells, running through this region, as we've said. But as they run through this region, they run in this pattern. They run through the primitive pit, and they run cranially. And when they run cranially, they are deposited to form a rod-like structure that is called the notochord. And this is the notochord. At this region, there is no mesoderm deposition. What you have is the formation of a specialized structure that is called the notochord. So this notochord will then replace the mesoderm that is formed between the ectoderm and the endoderm. So as soon as the notochord is formed, how this is an induction process for neurulation to occur, which is the formation of the nervous system, it will help to thicken the surface of ectoderm. So as soon as this notochord is formed, it's going to initiate the thickening of the surface ectoderm that is overlying the notochord. And this is where we now have the development of the neural plate. This will be discussed in our lecture on the development of the nervous system. So it's good for us to know that the development of this notochord is what will initiate the thickening of the ectoderm that is overlying the notochord. And this thickening will lead to the formation of the neural plate, which is the basis for the formation of the nervous system. So the final presentation that will be seen, we already know, is the formation of the prechordal plate, which is thickening of the endoderm that, of course, will be in contact with the surface ectoderm. So this thickening, of course, we say that it will help to create the craniocaudal alignment of the body. Then this is followed with the creation of the primitive streak. The primitive streak will be formed so that groove will be generated. As soon as groove is generated, there's going to be the creation of the path through which the surface epiplastic cells can move to form the 3 germ layers. And that will be the next event, which is the establishment of the 3 germ layers, which include the ectoderm, the intraembryonic mesoderm, and also the endoderm. Then finally, we have the formation of the notochord, which will help to initiate the process of neurulation, which is the development of the nervous system. So let's check our understanding of this lecture through the following questions. Let's describe the process of gastrulation. How does it occur? Also explain how the primitive streak, the primitive node, the groove, and pit are formed. Then the final question is, how is the notochord formed? I'll be expecting your responses in the comment section. So thanks for watching this video. Let's meet in our next lecture class.